Welcome to Pathophysiology, and today we are going to be lecturing on anemia, but we're going to start talking about background facts about blood first, and then we'll get into general information on anemia and specific types of anemia. So looking at blood, you have basically three different parts to blood because blood is a type of connective tissue. So in order for something to be a connective tissue, you have to have cells, you have to have a matrix, and you have to have fibers in that matrix. So we have red blood cells and we have white blood cells, and then we also have the plasma. And the plasma is the liquidy portion, the matrix portion, you might say. And then within that plasma are fibers, especially a type called fibrin. And fibrin is going to be important, and we'll talk about this a bit later. But fibrin works in what we call the coagulation cascade, which helps us to be able to form blood clots. So 55% of our blood is the plasma on average. And then we have 5 to 6 million red blood cells per milliliter and about 5,000 white blood cells per milliliter. And then we also have platelets, and platelets are also going to work for us as far as blood clotting is concerned. And so these little dots that you see in here, these are the platelets. So in red or pinkish, these are our red blood cells, and then these cells that have been stained purple, these are white blood cells. And so this just shows you a normal looking red blood cell and then normal looking white blood cells with some platelets scattered throughout. So when we talk about blood production, whether we're talking about white blood cells or red blood cells, we're talking about hematopoiesis. And this is going to occur in what we call myeloid tissue, which is in the red bone marrow. And that red bone marrow is found within different areas of our skeleton. So we have the axial skeleton as well as the girdles. And so these are going to be within the epiphysis of our long bones, such as the humerus and the femur, and also in the sternum and in the hip bones. So that our red bone marrow is going to have different stages of erythrocyte maturation. So we're going to be able to see immature red blood cells. We're also going to see reticular connective tissue within that red bone marrow. We have in the red bone marrow cells that are called hemocytoblasts, and these hemocytoblasts are going to help us to produce our red blood cells and our white blood cells. So the hemocytoblasts are going to become lymphoid stem cells, and those give rise to the white blood cells, also referred to as lymphocytes. And then we have myeloid stem cells, and these are going to help us to produce all the other blood cells. So you can see here, this is our hemocytoblast and our myeloid stem cell and our lymphoid stem cell. This lymphoid stem cell then becomes our lymphocytes, which are our T cells and our B cells. And then the myeloid stem cell is going to become red blood cells, and then our other white blood cells such as eosinophils, basophils, neutrophils, and monocytes. So we're going to really, for now, just look at red blood cells. We'll go over white blood cells in a separate video. So when we look at the red blood cell or the erythrocyte production, we're talking about the steps of erythropoiesis. So we're going to start with our hemocytoblast here, and then this becomes what's called a proerythroblast. So the hemocytoblast turns into a myeloid stem cell. The myeloid stem cell then has the ability to become any of those types of blood cells. But eventually what's going to happen is that myeloid stem cell is going to decide which type of blood cell it's going to become. And so we say it's committed. And so once it commits, we're going to then go through basically 3 phases in order to become an erythrocyte. And these are going to be important when we look at other blood diseases. So in phase 1, you have the early erythroblast, and what's happening is we're going to have a lot of ribosome synthesis occurring in there, because remember, ribosomes are going to help us to produce protein. They help us with protein synthesis, and the big protein we're making inside a red blood cell is hemoglobin. So then in step 2, we're going from this early erythroblast to the late erythroblast, and then we're actually turning into what are called normoblasts. And so during this process, we're accumulating tons of hemoglobin molecules, and so much hemoglobin that eventually what happens is that there's no room in this red blood cell, and the nucleus is going to be ejected. When that nucleus is ejected, we end up in phase 3 of erythropoiesis, and now we have what's called a reticulocyte. That reticulocyte is going to go through some changes in structure in order to become an erythrocyte. So an erythrocyte, if you remember, this is that biconcave disc red blood cell that is able to then pick up oxygen, which will bind with all of our hemoglobin. So again, our hemocytoblast goes to a proerythroblast, then that goes to the early erythroblast, and we go through our 3 phases, making a lot of ribosomes in phase 1, accumulating hemoglobin in phase 2, ejection of the nucleus to form a reticulocyte in phase 3, and then our reticulocyte becomes that mature erythrocyte. Now, we still have some reticulocytes that float around in our blood, but on average, it's only about 1 to 2% of our red blood cells. Now, when we want to increase erythropoiesis, this is all going to be dependent on oxygen levels. So if our oxygen levels fall, so let's say somebody has hypoxia for whatever reason, low oxygen levels in the blood, I don't know, it could even be you decide to go up to Big Bear to go skiing, and you're on the high slopes, which is above 8,000 feet. And so oxygen levels are going to be low. And when that happens, you have chemoreceptors in the kidneys that are going to be able to pick up these low oxygen levels. And this stimulates the kidneys to produce a hormone called erythropoietin. Erythropoietin then stimulates the red bone marrow to increase this process we just talked about, which is erythropoiesis. So now we're going to have an increase in our red blood cell count, which then allows us to pick up more oxygen. And now we have an increased oxygen carrying capacity in our blood. So again, the kidneys are triggered by hypoxia, and we don't have as much oxygen for the tissues, and so erythropoiesis is going to then allow us to increase the amount of oxygen by increasing the red blood cells. Now, erythropoiesis does require some very important things. So you have to have enough protein, fats, and carbohydrates in your diet. But in order to make these red blood cells, you have to have iron available because that's going to be part of your hemoglobin. You also have to have vitamin B12 and vitamin B9, which is folic acid, available. So the body stores iron, and so that iron is able to be used to make our red blood cells. So we have iron in hemoglobin, we have iron stored in the liver and in the spleen and in the bone marrow. So iron, when it is stored, inside a cell, it's going to be bound to a protein, and that protein complex is referred to as either ferritin or hemosiderin, depending on which cells the iron is stored in. If iron is circulating in our blood, not inside of a red blood cell, the iron is then also bonded to a different protein, and this is called transferrin, because trans would be to transport, and ferrin would be the iron. This is a picture of an erythrocyte, and remember we talked about the fact that it is a biconcave disc, and basically what that means is it dips in the middle and on the top as well as on the bottom. Now, one of the things about this shape is that it allows oxygen to diffuse into the red blood cell very easily, very quickly, as well as allowing it to diffuse out of the red blood cell. The other important thing about this shape is that an erythrocyte, it has to go through these really teeny tiny capillaries. And in order to go through these capillaries, and it's gone through these capillaries really fast, okay, as it's flowing through the blood, it's going to have to be able to go around a lot of curves, and it's going to have to be able to flex and bend. This is also why erythrocytes don't last very long. Your average erythrocyte is going to last about 120 days, and that's because imagine taking like a sponge and constantly twisting it and letting it go, and then folding it in half and letting it go, and twisting it and folding it in half and letting it go. How long is that sponge going to last? Not very long. And same thing happens with our erythrocytes. So this biconcave disc allows for folding of the erythrocyte. It also increases the surface area, and that allows better oxygen absorption. The membrane contains a protein called spectrin, and that helps to give the erythrocyte its flexibility. It doesn't have a nucleus, remember, because we spit that nucleus out in the third phase, and so our erythrocyte is anucleated. It doesn't have any centrioles, it doesn't have any organelles, and we need to get rid of all of those things because, first of all, erythrocytes don't go through mitosis, so it doesn't need a centriole, and we don't need the erythrocyte to have to worry about all these other metabolic processes, which, by the way, are going to typically use oxygen, and we don't want our erythrocyte using up the oxygen it's carrying. So there's no cell division, no mitochondria to generate ATP. Now, there is some anaerobic ATP production to keep that red blood cell alive, but we're not going to be using our oxygen to do this. We're filled with hemoglobin. There's about 1 million hemoglobin molecules per red blood cell. So that's about 97% of the cell's content. And so what's going to happen is our hemoglobin is going to bind to oxygen, and we're going to produce what's called oxyhemoglobin. And as far as the numbers are concerned, the red blood cell is going to be what's referred to as a formed element. And in females, we have between 4 to 5 million cells per cubic milliliter. And in males, we have about 5.2 to 5.8 million cells per cubic milliliter. This is a picture of the structure of hemoglobin. And so hemoglobin is made up of basically two parts. You have what's called heme, and then you have globin. If you look here, this is the heme molecule. And in the direct center of the heme molecule is iron. Iron and oxygen have a very high affinity, so the two are going to be able to bond very easily. Now, surrounding this heme, and you can see here, this is the heme. There are 4 heme per hemoglobin molecule. There is a globin, and that's what these kind of curlicue things are. Globin is made out of protein. And so you have these 4 globs of protein all bonded together, which is what we would call a quaternary structure of protein. And these proteins, this is referred to as the alpha 1 protein, alpha 2, and then you have a beta 1 and a beta 2 protein. When oxygen binds to this heme, it causes this globin protein to change shape. That triggers a reaction, which then causes the next globin protein to change shape a little bit more and exposes the heme, making it easier for oxygen to bind. And then this globin changes shape and the oxygen can bind. This globin changes shape, the oxygen binds. So when oxygen binds to one of these heme, we call the hemoglobin molecule 25% saturated. So you have an oxygen saturation of 25%. If you have oxygen binding to all four of these heme on the hemoglobin molecule, you're now at 100% saturation, or you have an O2 sat of 100%. So erythrocytes are dedicated to respiratory gas transport, and that also includes carbon dioxide. So hemoglobin reversibly binds with oxygen, and most oxygen in the blood is going to be bound to hemoglobin. So we talked about the fact that hemoglobin has the globin protein, 2 alpha chains, 2 beta chains, and then we have the heme containing the containing the iron that the oxygen molecule bonds to. And each hemoglobin can bond to 4 oxygen molecules. So when we're looking at red blood cells, there's all kinds of different testing that can be done to make sure that our patients are nice and healthy. And one of those things that involves red blood cells is what's called a CBC, or a complete blood count. So this tells us how many red blood cells we have. This also tells us how much hemoglobin we have. And of course, these colors are representing the fact that hemoglobin is a molecule that can change color. So for instance, if you have a hemoglobin molecule that has 0 oxygen bound to that molecule inside of our red blood cell, you look at that red blood cell and it looks much, much lighter in color. So you get this super light pinkish type of color. The more oxygen that bonds to the hemoglobin molecules, and so the more hemoglobin molecules even that you have, the darker in color your red blood cell is going to be. So this is an indication of how much hemoglobin I have inside my red blood cell, as well as the amount of oxygen that is bonded to it. Hematocrit is looking at the percentage of red blood cells that I have in my blood. So if oxygen is bound to hemoglobin, we refer to this as oxyhemoglobin. If we have oxygen that has diffused out of the red blood cell and is no longer bonded to the hemoglobin, we call this deoxyhemoglobin. And then you can have carbon dioxide attached to the hemoglobin. Now, it doesn't bond to the heme. Carbon dioxide actually bonds to the globin portion of our hemoglobin molecule. And if that's the case, we call this carbaminohemoglobin. And so typically, the red blood cell drops off oxygen at the tissues and picks up CO2 to take CO2 for a ride back to the lungs, where the red blood cell is going to release that CO2 and then you exhale that carbon dioxide out of your body. So again, red blood cells, again, don't last very long, maybe 100 to 120 days, but they are moving through the body. So in their lifetime, they travel about 750 miles, which is us going from LA to Albuquerque, New Mexico. Quite a distance. Old erythrocytes become rigid and they become fragile, and then they can fall apart and their hemoglobin degenerates. And dying erythrocytes are engulfed by macrophage. The heme and the globin are separated, particularly this happens in the liver, and iron can be salvaged and reused later. It is stored in the tissues. Remember, we talked about this as hemosiderin or ferritin, and then transported in the plasma attached to the protein as what we would call transferrin. So the heme, once that red blood cell's fallen apart, is degraded into what we call bilirubin. And the liver secretes bilirubin into the intestines as bile. Now, when we talk about the heme here, we've removed the iron from the rest of this heme to store it away for later. So this heme is just a waste product. We can't use this anymore. And so the liver turns it into bilirubin, and then bilirubin goes into the bile, and then from the liver, it's put into the gallbladder, from the gallbladder into the small intestine. And the intestines then take that bilirubin and they turn it or metabolize it into urobilinogen. And then that leaves the body in feces. And it is in a pigment called stercobilin, which is kind of that brownish color, which is why the feces is the color that it is. The globin is a protein, so it's going to be broken down into the individual amino acids, and then that's released into the circulation, and we can use that for building blocks for other proteins. All right, so red blood cell indices mean the measure of the size of a red blood cell as well as hemoglobin content. So we're going to talk about mean cell volume, or MCV. This is the average size of a red blood cell. Mean cell hemoglobin, which is MCH, the average amount of hemoglobin per red blood cell. And mean cell hemoglobin concentration, the average concentration of hemoglobin per unit of volume in an average red blood cell. So how much hemoglobin do you have in, or the concentration of hemoglobin do you have in your average red blood cell? All right, so complete blood counts, again, include white blood cells, red blood cells, percentage of white cells, so we're looking at neutrophils, basophils, eosinophils, all separately, and then the amount of hemoglobin and your hematocrit, which is your percent of red blood cells. And then the complete blood count also looks at your MCV and your MCHC. So your mean cellular volume and your mean cellular hemoglobin concentration. So looking at mean cellular volume, this is about the normal volume or normal size of a red blood cell. So we call this normocytic. If the red blood cell is smaller than it should be, this is microcytic. And then a larger than normal size red blood cell is macrocytic. If you don't have as much hemoglobin, the red blood cell isn't going to look as red. It's going to look more whitish, pinkish color, and we're going to call this hypochromic. If you're looking at a normal red blood cell, because it's that biconcave disc in the middle, it almost looks whitish if you're looking at it face on, and this would be normochromic. So macrocytic, red blood cells that are too large. Microcytic, too small. Normocytic, just right. Normochromic has the right amount of hemoglobin. Hypochromic, too little hemoglobin. Additionally, we can look at size variations in a sample of red blood cells. And so you may have a sample where all the red blood cells are the same size, but you may have a sample where the red blood cells all have varying sizes. So some are macrocytic, some are microcytic, some are normocytic, and we call this an isocytosis. If the red blood cells have weird shapes, I mean, they look like half moons or triangles or teardrops, this is poikilocytosis. So if you take a look at these red blood cells, they all are just about the same shape. Some of them may be a little oblong or strange looking, but that could be because of the movement that's occurring as the picture is taken. But notice these are pretty much the right color, the right shape, and see how they have the white directly in the middle when you're looking at it straight on. and this is showing you that biconcave disc thing going on. So if you're not seeing that, that's probably because the red blood cell is turning at the time the picture was taken. Another good shot of normal red blood cells. So if you're looking at these red blood cells here, what would you call these? Are these normocytic, macrocytic, microcytic? Now this is a white blood cell, probably a basophil. So if I tell you that these here and here are normal size, then what are these? And if you said macrocytic, you would be absolutely correct. Now this is an interesting picture right here. This is a red blood cell that is losing its nucleus. All right, so you're looking at hypochromic microcytic anemia. So notice these red blood cells. They have a much larger region here in the middle of light color, and that's because they don't have as much hemoglobin as, let's say, this red blood cell here. This red blood cell is also smaller than it's supposed to be, so that makes it microcytic. And most persons at risk for this are going to be like children and women in their reproductive years, and this is mostly because of iron deficiency. So if I don't have enough iron in my system, I will have what's called iron deficiency anemia, and I can't make enough hemoglobin. And same thing that you see here. This is going to be hypochromic. This is definitely hypochromic. This is pointing to a little platelet here. What do you think these guys are? By the way, sometimes you'll see red blood cells piled on top of each other like this. I don't know if you've ever eaten the candy Rolos, and you have these little chocolate caramels that can stack on top of each other. That happens with red blood cells. They start stacking on top of each other, and they are actually called a Rolo as well, except it's not spelled the same way. It is a French term, and you can see right here these are stacking on each other, and that is a Rolo, R-O-L-L-E-A-U-X. So here again, you have a macrocytic red blood cell. Now here's some red blood cells that have weird shapes. What do we call those strange, unusual shapes, as well as having macrocytic red blood cells? Hypochromic microcytic, and then you can look through here. Oh, here's a really cool Rolo right there where they're all stacked on each other. But you can also look at the strange shapes. There's a teardrop shape here, another weird shape here. What does anisocytosis and poikilocytosis mean? Make sure you know those. All right, so let's talk about anemia, some just general information. An means without, emia in the blood. So this is a reduction below normal in hemoglobin or red blood cell number. Hemoglobin levels fall below the normal average, and the normal average for men are about 14 to 18 grams per deciliter, and for females about 12 to 16 grams per deciliter. So on average for men about 16 grams per deciliter, 14 grams per deciliter for females. The symptoms of anemia, because think about this, I don't have enough hemoglobin, or I don't have enough red blood cells in my system, and so I can't carry enough oxygen. So what are the symptoms for low oxygen or hypoxia? So pale skin, pale mucous membranes, jaundice if you have hemolytic anemia. And we'll talk about this, but hemolytic anemia basically means we have hemolysis of red blood cells. They're exploding. So I'm releasing that heme into the system. Tachycardia, breathlessness, dizziness, fatigue, all symptoms of anemia. And then we're going to talk about specific types of anemia now, and 3 ways to become anemic: lose too much blood, destroy too many red blood cells. So there could be what we call extracorpuscular reasons, and a corpuscle is the old-fashioned name for red blood cells. So these are from a source outside the red blood cell. Destroys extrinsic to the red blood cell are usually, disorders extrinsic to the red blood cell are usually acquired. And then intracorpuscular reasons, this is due to a defect within the red blood cell. So intrinsic red blood cell abnormalities are usually inherited. And then you make too little blood. So it's either because there's too few building blocks to make your red blood cells, or just too few erythroblasts in order to produce those erythrocytes. All right, so let's talk about the first of the 3 ways to become anemic, which would be loss of blood due to hemorrhage. And a couple of things that you need to know is that typically this is due to a trauma. So some kind of trauma is going to occur, and you're going to have acute blood loss. Now, you can also have blood loss that's chronic, so we'll talk about that in just a second. But with acute blood loss, at first, hemoglobin levels are typically going to be normal. Now, this doesn't mean that the person, just cut their femoral artery and they're hemorrhaging to death. This is going to be a slower type of traumatic blood loss or blood injury. And after 2 to 3 days, you're going to see more reticulocytes than normal because the body is trying to increase erythrocyte production. And then you can also have chronic blood loss. And that chronic blood loss, again, is going to be a slow chronic bleed that somebody has. Like, for instance, you can have a person who is taking too many NSAIDs. And as you take those NSAIDs over time, you're actually destroying the intestinal lining, because NSAIDs are very acidic and they can potentially poke little holes in the walls of the intestine, and that could lead to a slow chronic bleed. And if that happens, your patient also is going to see iron deficient anemia occurring. So reticulocytes are these guys right here with this increased staining here in comparison to red blood cells, normal erythrocytes. And so these are immature red blood cells. They usually typically compose about 1% of the blood cells overall, and they have more of a convoluted shape. They're about 8% larger than the mature red blood cells, but they still don't have a nucleus. Instead, that blue stuff that we saw, that's kind of a reticular mesh-like network of ribosomal RNA. Because remember, these cells are still trying to produce hemoglobin, so we need a lot of those ribosomes. So you're going to see more reticulocytes in somebody's blood who has had blood loss. So another way is to destroy too much blood, and this could be for extracorpuscular reasons. And one of those is a type of red blood cell destruction called hemolytic anemia. So hemolytic anemia is a condition in which there's premature destruction of the red blood cells, or hemolysis. This is usually associated with, again, high count of reticulocytes. And then you also have an increase in the amount of LDH in the blood. LDH is lactic dehydrogenase, and this is an enzyme that dying red blood cells release. You will also have low blood haptogens. These are proteins that bind hemoglobin from dead red blood cells and carry it away for disposal. Then, because these red blood cells are going through hemolysis, we're going to have an increase in the heme in our system, which we refer to as blood bilirubin. Some of the causes of hemolysis: genetic red blood cell defects, immune problems such as autoimmune diseases. And these autoimmune diseases, our body attacks its own red blood cells, and we have what are called hemoglobinopathies such as sickle cell anemia, thalassemia, which we'll also talk about later. Then you can have mechanical destruction of the red blood cells, so some kind of trauma can cause this hemolytic anemia. Infection, so malaria is one. Clostridium, which releases a toxin, and there's other bacteria that can do the same thing, causes hemolysis of red blood cells. Snake bites are a big deal as far as hemolysis is concerned. And then certain drugs that we can take, legal or illegal, can cause hemolytic anemia. Symptoms of hemolytic anemia: dark urine because there's excess blood being destroyed and cleared out by the kidneys. An enlarged spleen, so that splenomegaly is basically the same thing. The spleen is catching those Then you also have an enlarged spleen or splenomegaly because that spleen is trying to get rid of all of those dead red blood cells. Fatigue because I don't have enough oxygen. Fever if there is some type of bacterial infection going on. Pale skin, rapid heart rate, shortness of breath, that jaundice from the bilirubin. Chills can occur because my red blood cells help to carry heat, and so I don't have as much heat carrying capacity. There's another type called microangiopathic hemolytic anemia, and this is physical trauma to the red blood cells and schistocytes form. And so the big deal here is that you have these red blood cells and they're being snagged by these fibers here, which are proteins in the red blood cells called fibrin. So what happens is you have some kind of damage, some kind of trauma to the endothelial lining of our small blood vessels. And because of that, we have fibrin that's being deposited as well as platelet aggregation in that lining. So now our red blood cells are traveling through these damaged vessels with all this fibrin, and the red blood cells are getting caught and stuck in this fibrin, and the red blood cells, now imagine it's this wave of blood that is flowing through like a fast-moving stream, and your red blood cell gets caught onto this sticky fibrin, but the stream is still pulling on that red blood cell, and what it does is it rips it off the fibrin and causes that red blood cell to actually change shape and form into what we call a schistocyte. And so this is what we're looking at here. Okay, all these funky, weird shapes of our red blood cells, these are schistocytes. These are red blood cells where chunks basically have been taken out of the red blood cell. The red blood cell has been torn, and eventually this red blood cell is going to die. It doesn't function well. So these are fragments of red blood cells, and this can be caused by what we just talked about, and what this is very often called as DIC, disseminated intravascular coagulation, which is where you get that clotting on the walls of the small vessels. And these blood clots can reduce or block blood flow and damage organs as well. Sometimes those schistocytes actually look like little triangles, and so they call those triangulocytes. So causes of this could be artificial heart valve, malignancies, obstetric complications, sepsis, trauma, snake bites. Autoimmune hemolytic anemia. There are two types. We call this warm autoimmune hemolytic anemia, and then there's cold. So with warm, the antibodies are being made because this is autoimmune. So autoimmune diseases are all about our body producing antibodies that are going to destroy something by mistake. And so antibodies are made to destroy red blood cells, and this happens at body temperature. So this is an IgG type of antibody. In cold autoimmune hemolytic anemia, antibodies can become active when parts of the body are exposed to lower temperatures, such as your hands and your feet, and this is usually an IgM or complement type of antibody made. So for warm autoimmune hemolytic anemia, it occurs at body temperature. IgG is what's made most often, sometimes IgA. It attaches to red blood cells, leaving what's called the Fc portion exposed. Now this Fc region is grabbed onto by macrophage in the spleen. And the macrophage basically take a bite out of the red blood cell, and it becomes that spherocyte. So the spherocytes aren't very flexible, and then they can be destroyed by the spleen, and unfortunately, if there's a lot of them, you end up getting splenomegaly. So this is the red blood cell, and these are the antibodies that are attached to that red blood cell. This is the Fc portion that is exposed. And then along comes your red blood cell, which can attach to this Fc portion. And what happens is it ends up taking a little bite out of your red blood cell, and now your red blood cell looks all funky and distorted, and you have a spherocyte. Actually, here's a pretty decent picture of one that had a little chunk taken out there. Cold autoimmune hemolytic anemia is 60% of it caused by infectious mononucleosis, although some of it is idiopathic. It is associated with cold exposure, could be also with infection, and we call the IgM the cold autoantibodies. And this is going to be the same thing as warm, where you have IgM now attached to the red blood cells, and then our macrophage can come in, attach to this, and take a chunk out of the red blood cell. And you can see a lot of agglutination occurring here with these red blood cells, but like here's one where a chunk was taken out. This one looks like it was bitten off of here. So there's a few that have parts taken out that become a spherocyte. So sometimes we can do what are called antiglobulin tests, and it's basically the same thing where we put antibodies in and they bind to these antibodies that are attached to our red blood cells, and then you get what's called agglutination. You get blood clotting forming. This shows you that the patient has these autoantibodies in their system. And usually the only treatment for this is like corticosteroids to calm the immune system down and hope that it will stop producing these excess amounts of antibodies. Another way to become anemic is to destroy too much blood by intracorpuscular methods. And so this includes, for instance, sickle cell anemia. And things you need to know about sickle cell anemia: this is what's called a hemoglobinopathy, and this is a defect in our hemoglobin. It is a single amino acid substitution in the beta chain of the hemoglobin. And so basically what that means is there is a point mutation in the DNA. That means one of the ATGCs are in the wrong place. And because of that one mutation in the DNA, there is one amino acid that gets put into the hemoglobin globin portion of the beta chain and makes that beta chain the wrong shape. In order for this to happen, the person could be heterozygous or homozygous. And sickle cells are very fragile. They burst very easily, and then they can be stuck in blood vessels as well. So here's our hemoglobin again, and so one of these chains has an incorrect shape. So again, you have a point mutation that forms the beta chain, and what happens is there's a substitution for the amino acid valine being put in instead of glutamate. And now the chain is the wrong shape, and so now we give it the name hemoglobin S and not just hemoglobin B anymore. The problem is also that you can't pick up oxygen as well. So the person's blood has lower oxygen levels, but the red blood cells become that sickled shape, which gives them their increased fragility, increase in the ability of them getting stuck in blood vessels, because this pointy end can stick into the walls of the blood vessels, they can stick into each other, and then you get clogs going on. And if you get a clog in a blood vessel and blood can't continue to flow, the issue is on the other side of that clog, there isn't enough oxygen. And when cells don't get enough oxygen, they let you know that oxygen levels are critically low by releasing chemicals that stimulate pain. And so our person who has a lot of these blood clots going on in their body are going to have severe pain issues. So this is showing you some of the irregular shaped or sickle-celled red blood cells. And so you can see in the top picture just normal red blood cells cruising through a blood vessel, but when the cells in the bottom picture are now sickled, they start to get stuck in the wall of the blood vessel as well as stuck in each other. So sickle cell anemia has normal hemoglobin A, but abnormal hemoglobin B, so we call this hemoglobin S. And this is that single base mutation, changes the one amino acid. And so with hemoglobin S, I can't pick up as much oxygen. I have a distorted, less flexible shape. And there's a much nicer looking picture of a sickled red blood cell. So deoxygenation is a big problem for these patients. And this just shows you how it can get stuck in the areas where they're supposed to flow easily and bend. Because remember, I told you red blood cells need to be very flexible and they need to be able to bend and change shape to get around corners. And sickle cells can't do that very well. Also, sickle cells only have a lifespan of about 20 days, so you have this chronic anemia that occurs. And because I don't get enough oxygen, because my sickle cells can't pick up as much oxygen, I can't get oxygen to the skin, to certain organs, and things can start to break down. So I can get lesions inside and outside the body. There's also something called sickle cell crisis, and this is where you get an acute sickling episode that blocks blood flow, and it can pose a huge widespread threat to our body because there's ischemia that leads to organ damage, and then pain is due to lack of oxygen, which then leads to necrosis. And that necrosis can be in multiple organs, including the bones. And these are the sickled cells within the spleen. Clinical findings of sickle cell anemia: about 8% of African Americans are heterozygous for sickle cell anemia. The severity of sickle cell anemia can vary. There's chronic hemolysis, vaso-occlusive disease, and increased infection, and then autosplenectomy, which means that the spleen can just die on its own. Treatments prevent triggers, and also vaccinations and transfusions for sickle cell anemia.